So today I am going to discuss about the application of plant tissue culture. What is the application of plant tissue culture? So try to know what are the uses or application of plant tissue culture. That is uh, means this is the slide that is the regarding application of plant tissue culture. Okay, so try to know production of different useful compound. What are the different useful compound? That is the alkaloids or saponin. You know what is secondary metabolites or chemical that is secreted by plant to protect the plant from different type of germs like bacteria, fungus, virus, or insects, or animals, or rodents. Means a chemical that is secreted by plant by itself, but main objective for the production of secondary metabolites inside the plant is just to protect the plant from itself, from different type of germs. Type of germ means bacteria, fungus, virus, rodents like rat, mice, and then insect that can damage the plant leaf, damage the plant fruit, damage the plant stems. So an animal also, means main objective for the production of secondary metabolite is just to production of different type of what, different type of bacteria, production from different type of germs, bacteria, fungus, virus. So that is the main objective for production of the secondary metabolites. Now human has utilized these secondary metabolites, that is used for different treatment of different type of disease. So please join the class on the time. I'm requesting all the students, please join the class on the time, uh, because if you left the class, there is no alternative for biotech. Okay, so secondary metabolite production inside the plant through molecular farming. Okay, so what do you mean by molecular farming? Means creation of transgenic plants, and then to enhance the production of secondary metabolites. It can have useful compound, it can be steroids. Okay, it can be a steroid, it can be anti-tumor compound. So it can have chemicals that used for treatment of different type of disease. So these are the different chemical compound that is secreted by plant itself through metabolic activity. So scientists want to optimize the production of this secondary metabolite for utilization of that secondary metabolite compound in the plants. After extraction of secondary metabolite, production inside the plant utilized for different type of making different type of medicine, drugs that can be used for treatment of different disease. Okay, so main focus is production of secondary metabolite. Okay, what are secondary metabolite? A chemical compound that is secreted by plant to protect the plant itself from different type of disease germs like bacteria, fungus, virus, insects, harmful insects, then rodents, then animals. Means but just for survival of the plants they are producing. But human people, scientists utilizing this secondary metabolite for treatment of disease. Means chemical secondary metabolite that is synthesized by plant. So what is the main objective? Production of transgenic plant that can produce huge quantity of secondary metabolites like alkaloids, saponin, steroid, anti-tumor. Anti-tumor means, have you seen Catharanthus rosea plant? Inside the Catharanthus rosea plant, there is a anti-tumor compound. What is anti-tumor compound? Vincristine, Vincristin, vincristine and vinblastine. These are used for treatment of cancers. Okay. Now, a biopesticide. Pesticide means just a chemical secreted by the plants that use as a biopesticide to kill the pest insects. Main focus is just to kill the harmful insect that is going to damage the plants. Food additives that preserve the food that is synthesized by the plants, and chemicals that used for different treatment of the disease or used for making different chemical preservative in medicine. So that is the main objective for plant tissue culture. Means through plant tissue culture we multiply the plant and then insert the useful gene. That gene enhance the production of secondary metabolite. Means first creation of transgenic animals through recombinant DNA technology, and then after creation of recombinant cells of the plant, we multiply that cell through plant tissue culture, and then we grow the large number of plant so that we can produce huge quantity of that chemicals inside the plant itself. That concept is known as molecular farming. Means a particular chemical is going to synthesize in particular transgenic plants just to optimize the production of that particular secondary metabolites that can use for treatment of disease. Okay, so that is the concept of molecular farming. Next objective is production of virus free plants. What is virus free plant? Means through plant tissue culture we multiply the plant, and what is the source of that plant sample? That is the apical meristem. What is that? Is the apical, just see the arrow cursor, that is the apical meristem. You know what is apical meristem? If you observe the plant, growing plant terminal end, either the root or a stem, then it has terminal portion that have capacity to divide and multiply. We have already discussed these things in tissue chapter in biology. Means that has ability to divide and multiply. That is that is free from virus. Why? Why it is free from virus? Because at terminal end, no xylem and phloem reach up to that terminal, and the pathway to, through which virus is traveling is only xylem and phloem. So if you see the terminal end of the plant stem or leaf, then you find there is no supply of pathway of xylem and phloem. You know what is the function of xylem? Xylem transport the water and minerals from root to terminal end. Yes, आप बताइए, कुछ कंफ्यूजन है तो बता, पूछ सकते हो. Okay, so the main pathway through which the virus plant part affected by virus is xylem and phloem, but this xylem and phloem is not reaching up to terminal apex portion of the stem or leaf. So that's why there is no chance of infection of that terminal end, and we cut the 1 millimeter length of this plant that is free from virus and use as explant for multiplication of the plant. So first point, there is no reaching of the xylem and phloem up to the terminal end. Second point, there is a high concentration of synthesis of endogenic oxygen, and the oxygen, high concentration of oxygen prevent the growth of virus. High concentration of production of oxygen prevent the growth of virus. So this is the reason endogenous oxygen production at the apical meristem portion that can utilize, that is we are going to utilize for multiplication of plant, for the rapid multiplication of plant that is free from virus. That's why we get the virus free plant, and we do all the process of plant tissue culture. It can be of ornamental plants, it can be of vegetable plants, it can be of fruit bearing plants. We get the virus free plant and we rapid multiply that type of particular variety of plants, particular genus of the plants through plant tissue culture. So this main two reason is they know reaching of xylem and phloem up to terminal end of apical meristem. That's why there is no chance of infection. Second point, huge concentration of oxygen. Huge concentration of oxygen that prevent the growth of what? The virus. That's why we select the apical meristem and we do all the process. We do all the process of plant tissue culture in aseptic condition. What do you mean by aseptic condition? Means the environment where we are doing that practical multiplication of that plant at commercial scale, at large scale, in the germ free environment, means inside the laminar airflow, inside the chamber, means that is free from germs, spore or germ, bacteria, fungus, virus. So we are expecting that we are getting the virus free plant through plant tissue culture. But if we are growing the plant through reproductive pathway, there may be chance of presence of virus in growing plants. So generally we prefer the plant produced by plant tissue culture. And why we prefer? Because it is virus free plant and we get large number of that plants within a very short period of time. Okay. Next, what is micro propagation? It is a method for rapid multiplication of plant through plant tissue culture. It is a method for rapid multiplication of plant through plant tissue culture. In that case, we do not develop the uh, callus. What do you mean by callus? Callus is undifferentiated growth of the plant cell. That we cannot say it is the root, we cannot say it is the stem, but from this explant there is a proliferation of large number of shoot. If we supply the suitable media and that contain high concentration of cytokinin, then there is a rapid multiplication, rapid growth of the stem from the same explant. So individually we cut it and then we treat as individual plant through micro propagation. So where we apply the micro propagation for the multiplication of forestry, for the multiplication of forestry, fruit crop bearing tree, vegetatively propagated plant tree, plantation crop. Plantation crop means that we use for um, fodder purpose or for any purpose, means you can use the tea also, you can use for fodder purpose, you can use for any industrial purpose. And essential oil means that essence means perfumes, that plant produce the perfumes like chemicals. Okay, peppermint. Okay, so that is used for the rapid multiplication of plant through micro propagation. So micro propagation, it is a technique through which we rapidly multiply the particular variety of plant, particular genus of the plant for commercial point of view. Means if you want, you have to multiply the plant at large area. How can you multiply? With the help of this micro propagation, and we get the quality of the plant. That is the same quality because we are going to multiply the plant from somatic cell, not reproductive cells. That's why we prefer the micro propagation. Propagation means we do not pass this explant into the callus. Generally, directly we proliferate the formation of a stem. How it is possible? With the help of addition of huge quantity of the cytokinin plant growth regulator, because this cytokinin proliferate for rapid multiplication of stem portion only, and then individually we cut whatever the number of stem proliferate from the stem explant from proliferation of a stem, that is the from the explant, then we cut individually each one and we get the individual plant. We change the media and we grow the root system. So first is secondary metabolite production, second is virus production, third is embryo micro propagation, and next is anther or pollen culture. What is the application of anther and pollen culture? Means you collect the anther. First you collect the anther from the flower part, that is the male reproductive part, and then we multiply the cell inside the plant tissue culture medium. Multiply that cell in plant tissue culture medium. So just for getting haploid, do you know what is haploid? पता है आपको haploid क्या है? Haploid, that is the half number of chromosome. 2n is equal to कितना होता है? Suppose in rice 2n is equal to 16, तो n कितना होगा? N is, if 2n is equal to 16, then what is the number of n? Reply, give me reply. कोई बता सकता है? अगर 2n is equal to 20 है तो n कितना आएगा? n, अगर 2n is equal to 20 तो n is the haploid number of chromosome, that is 10 हो जाएगा. तो ये मैं बता रहा हूं कि जनरली द एग and pollen grain nucleus, that is the cell, that is the haploid. Okay, have you heard about endosperm? Endosperm का क्या haploid level होता है? क्या होता है बताओ?
3n.
होता है हाँ? Yes,
3n.
3n, yes. So generally try to know 2n generally in case of somatic cell and n generally in case of reproductive cell, because after meiosis means reductional division, number of chromosome reduced to, that is in case of pollen grain. In case of the egg, you will get the half number of. So just for haploid plant production, and after colchicine treatment, colchicine is a chemical that will fuse two haploid and we get the homozygous line. Okay, so colchicine is a chemical that fuse the two haploids set and make a n plus n. Okay, so generally we get a homozygous similar type of plant population through anther culture, pollen culture. How can you do this anther culture? Means you collect the anther of desired quality of particular plant flower, and then we multiply that pollen grain inside the plant tissue culture media, and then we develop the plants. So that plant is the haploid. And if we use colchicine, a chemical that will make the diploidization making of haploid and haploid into 2n, then we get a homozygous line. So this is the objective for production of haploid plant, homozygous line for plant breeding, means production of new variety of the plants. So these are the application of plant tissue culture, means production of secondary metabolites, secondary production of virus free plant, third multiplication of the different variety of the plant like forestry plant, fruit crop plant. Yes, plantation. हाँ कुछ पूछ रहा है आप लोग?
सर ये colchicine है जो ये cell poison
chemical that is obtained from the plant Colchicum autumnale. ये नाम है प्लांट का, आप देखेंगे, आप गूगल में जाके सर्च करेंगे, इट इज यूज फॉर फ्यूजन ऑफ टू हैप्लॉइड. ओके, कॉल्चिसिन इट इज ए केमिकल फॉर डिप्लॉइडाइजेशन, मींस अगर दो हैप्लॉइड है, हैप्लॉइड प्लस हैप्लॉइड दोनों को जोड़ देंगे, अगर दोनों डिफरेंट वैरायटी है, हाँ यस, दोनों अगर
ये सेल
डिफरेंट,
पॉइजन है।
हाँ हाँ हाँ, ये आपको प्लांट से मिलता है, डिफरेंट प्लांट से, लेकिन उसको एक्सट्रैक्ट करके जस्ट फॉर एक्सपेरिमेंटल पर्पस दे वी आर गोइंग टू फ्यूज द टू हैप्लॉइड सेट ऑफ द टू डिफरेंट वैरायटी, ओके, जस्ट टू मिक्सिंग द जीन of the two different variety of the plants, okay, means we are going to fuse and to proceed for diploidization, diploid formation, haploid plus haploid equal to होता है diploid, मतलब हाफ नंबर ऑफ n प्लस n इक्वल टू 2n, तो 2n अगर ऐसे नहीं होगा, अगर छोड़ देंगे तो नहीं जुड़ेगा, जब तक उसमें केमिकल नहीं डालेंगे तब तक उसमें पेयरिंग नहीं होगा, ओके, सो जस्ट आर्टिफिशियली इंड्यूस द फ्यूजन ऑफ टू हैप्लॉइड, अगर n प्लस n है तो इसको हम नहीं बोलेंगे कि ये डिप्लॉयड है, इट इज नॉट ए डिप्लॉयड, 2n हो जाएगा तब होगा व्हेन वी पुट द colchicine of chemical that used for diploidization, ओके, समझ में आया?
सर ये
नेक्स्ट है,
कॉल्चिचिन है जो,
हाँ बोलिए।
सर ये सेल साइकिल को कौन सी स्टेज में अरेस्ट करता है, सेल डिवीजन को?
सेल डिवीजन देखिए यहाँ पे सेल डिवीजन में जब जनरली हम कलेक्ट करते हैं ये आफ्टर मियोसिस वी गेट द हैप्लॉइड, ओके, एंड आवर ऑब्जेक्टिव इज जस्ट टू प्रोड्यूस द hybrid variety of the particular plant that have high vigor, that have high characters, means that have capacity to produce huge quantity of grains, huge quantity of secondary metabolite, huge quantity of particular cereals. In that situation, if we, after completion of meiosis, if we are getting the haploid either pollen grain or egg, okay, in that condition means after production of haploid set, after complete meiosis we collect the haploid cell, and then after collection we mix along with these chemicals, then we left it for sometimes 10 minutes 20 minutes, then again we put in the media, and then it will produce the diploid, means character coming from two different plants, two different variety, and then we get the character of both the plants, good quality good character of both the plants. तो ये ऐसा कुछ नहीं है, आफ्टर कंप्लीशन ऑफ मियोसिस वी कैन फ्यूज द टू हैप्लॉइड सेट ऑफ द क्रोमोसोम जिसको टू गेट द डिप्लॉयड, or if you want to do research breeding purpose, means haploid variety, means just to do any creation of uh, transgenic plant or hybrid variety of the plant, in that case we prepare a line of haploid plants. So just for experimental purpose we do this process so that we can produce best variety of the plants, okay, or it can be plant of the vegetable, it can be plant of or fruits, it can be plant of essential oil. Do you know essential oil? Oil that have perfumes, that uh, create a perfume used for perfume preparation, that is known as essential. Plantation crop आपको पता है, means we can utilize this technique for all the type of crops. It can be vegetable, it can be a fruit, it can be essential oil, it can be a plantation crop आपको हो सकता है, टी को भी बोल सकते हैं, कॉफी को भी बोल सकते हैं, ठीक है ना, जनरली प्लांटेशन का उसी को बोला जाता है, और आपको सेकेंडरी मेटाबोलाइट प्रोडक्शन करना है तो उसके लिए नेक्स्ट एप्लीकेशन दैट इज द प्रोडक्ट्स एम्ब्रियो रेस्क्यू, एक होता है एम्ब्रियो रेस्क्यू। व्हाट इज एम्ब्रियो रेस्क्यू, किसी को पता है? एक बच्चा किसी ने क्लास छोड़ा है, अभी कौन छोड़ दिया है, अभी 7 हो गया, so try to attend the class on time, everybody is watching you from the RO चंडीगढ़, ठीक है, तो प्लीज ट्राई टू अटेंड द क्लास ऑन टाइम एंड लेफ्ट, बिकॉज एवरीथिंग इज रिकॉर्डेड, कितना देर आपने क्लास में अटेंड किया ये रिकॉर्डेड है, ठीक है, तो ये मत सोचना कि मैं आ गया चला गया, ऐसा नहीं होता है, आप 1 मिनट रहेंगे 2 मिनट रहेंगे 30 मिनट रहेंगे, उतना टाइम आपका रिकॉर्डेड रहेगा। नेक्स्ट इज एम्ब्रियो रेस्क्यू। व्हाट इज एम्ब्रियो रेस्क्यू? इट इज द टेक्निक फॉर सेविंग द प्लांट देयर इज ए एम्ब्रियो एबॉर्शन। एम्ब्रियो एबॉर्शन मतलब कि ड्यू टू क्रोमोजोमल इम्बैलेंस, अगर क्रोमोजोम का नंबर 2n इक्वल टू अगर 20 है, अगर 20 की जगह 23 हो गया या 24 हो गया, दैट प्लांट विल नॉट ग्रो, हो गया, इंडोस्पर्म इम्बैलेंस हो गया, ठीक है ना, तो दैट प्लांट विल नॉट ग्रो. आफ्टर फर्टिलाइजेशन देयर इज जायगोट फॉर्मेशन, बट दैट जायगोट विल degrade. In that situation, what you have to do? In early stage, you will cut the that zygote, early embryo, जिसको बोलते हैं अर्ली एम्ब्रियो. अर्ली एम्ब्रियो क्या मतलब क्या होता है? मेल एंड फीमेल गैमीट इज हैव फ्यूज्ड, मेल एंड फीमेल गैमीट हैव फ्यूज्ड, and then it formed the zygote. Now zygote has formed. After few days, this zygote will degrade. Before degradation of the zygote, due to chromosomal imbalance that is happening, due to chromosomal imbalance that is going to happen, in that situation you cut, as a scientist you cut that embryo and put in the plant tissue culture media, so that you can multiply that plant, because that if there is no formation of the seed from any plant, it become in the category of endangered species, and then finally into become extinct हो जाएगा. सो दिस इज द एम्ब्रियो रेस्क्यू. इस एम्ब्रियो रेस्क्यू मतलब आप एम्ब्रियो को बचा रहे हैं. क्यों बचा रहे हैं? जस्ट टू मेंटेन द प्लांट पॉपुलेशन ऑफ दैट प्लांट वैरायटी. कुछ ऐसा प्लांट है जिसका सीड नहीं बन पा रहा, मीन्स रेयरली देयर इज अ फॉर्मेशन ऑफ सीड. जब तक सीड नहीं बनेगा तब तक प्लांट मल्टीप्लाई नहीं होगा. लेकिन अल्टरनेटिव क्या है? प्लांट टिश्यू कल्चर. वेरी गुड. मीन्स यू कैन मल्टीप्लाई द प्लांट फ्रॉम द एम्ब्रियो. You can use the embryo cell or tissue and then put in the media. In earlier stage, after formation of zygote, just little bit development of that zygote, that is known as early embryo, and you cut that early embryo in earlier stage and put in directly into the plant tissue culture media. Then there is a multiplication of that cell. Means, and from that cell you can multiply the plant, large number of plant you can get, and then maintain the plant population. That is known as embryo rescue. आपको पता है embryo rescue क्यों होता है? पता है embryo rescue क्यों होता है? Due to chromosomal imbalance. क्यों होता है ये? Due to chromosomal imbalance. Means imbalance. Suppose 2n is equal to होना चाहिए 30. 2n is equal to क्या हो रहा है? 31 हो रहा है, 32 हो रहा है, 33 हो रहा है, ऐसा होता है. या 2n is equal to 60 हो गया. In that situation, plant will not develop the viable embryo, okay, and there is no formation of seed. In that situation, you have to cut the embryo from the plant and put in the plant tissue culture media. Now this is the explant. I have already discussed this explant, okay, and you try to know what type of micropropagation. Just see in this diagram, micropropagation. You see the step, establishment हो रहा है, धीरे धीरे इसमें डेवलप रूट स्टेम प्रोलिफरेशन हो रहा है. समझ में आ रहा है? ये मीडिया दिख रहा है आपको, एस्टेब्लिशमेंट एक्सप्लांट इन द कल्चर मीडिया एंड सस्टेन द प्लांट डिवीजन. दिख रहा है आपको? ये दिख रहा है. दे आर गोइंग टू ट्रांसफरिंग द एक्सप्लांट इनटू अ सुटेबल मीडिया. मींस आफ्टर सिलेक्शन ऑफ द एक्सप्लांट, they are going to put the explant in the suitable inside the laminar air, inside the laminar after transferring. Means इसको कहते हैं incubation. Means putting the explant in the plant tissue culture media that is present inside the jam bottle, and we cover it. ठीक है ना, उसको कवर करते हैं और कंप्लीटली सेलोटेप की तरह आता है, पैराफिन से मेम्ब्रेन से उसको कंप्लीटली क्लोज कर देते हैं, सो दैट एक्सटर्नल फ्रॉम द एक्सटर्नल साइड देयर इज नो चांस ऑफ इन्फेक्शन. तो आपको ये नहीं होना चाहिए कि यहाँ पे चांस ऑफ इन्फेक्शन हो. देन यू विल डिसाइड इन व्हाट यू हैव टू प्रोड्यूस. आइदर फर्स्ट यू प्रोड्यूस द स्टेम and then root. तो फर्स्ट स्टेम बनाना है तो साइटोकाइनिन कंसंट्रेशन ज्यादा होगा. आफ्टर फॉर्मेशन ऑफ कैलस, आई एम टॉकिंग अबाउट आफ्टर फॉर्मेशन ऑफ कैलस. इन दैट सिचुएशन मीडिया कंटेन इक्वल क्वांटिटी ऑफ ऑक्सिन एंड साइटोकाइनिन. बट second step, that is formation of stem, that is media contain high concentration of cytokinin and low concentration of organ. If you want to produce the proliferation of root, In that situation, they reverse. Media contain low quantity of cytokinin and high quantity of oxygen. Try to remember 50-50 concentration callus formation. मतलब equal quantity of oxygen and cytokinin callus formation. So and high concentration of cytokinin root stem formation and high concentration of oxygen, that is root formation. So root formation we proliferate. Step 4 means shoots are transferred to a growth media with relatively high oxygen concentration. समझ में आ रहा है? ये तभी ये root बनेगा, otherwise there is no formation of root. Try to know, this is the jam bottle and that is the media, and in the inside that media we put the high concentration of oxygen cytokinin ratio. So you will find this thing in banana plants, that is generally used for commercial point of view. And now the next step you will put in the uh, greenhouse and glass house, and then field condition. So the rooted shoot are potted up and hardened up by gradually decreasing the humidity. Means you are going to reduce the moisture content of the environment inside the glass house or greenhouse. Okay, so these are the plants are fully prepared. Now you put in the soil. Okay, and then there is necessary many young tissue culture plant have no waxy cuticle to prevent the water loss. Okay, so in that situation we try to hardening means gradually acclimatization, means from lab situation to environmental, shifting from lab situation to environmental situation, that is known as hardening. Hardening means development of xylem and phloem, only then it become autotroph. In earlier stage in the lab it is prototroph, now it is not a autotroph because there is no proper development of xylem and phloem so that it can do the process of photosynthesis, because they are not getting the water and minerals from the soil. So generally the process that hardening means development of xylem and phloem to become self-sufficient for the process of photosynthesis. So that process, that is the development of xylem phloem, development of root and fruit formation, and development of xylem phloem is known as acclimatization. Means shifting a plant from lab condition to field condition. First you shift in greenhouse, then glass house, then field condition. So this is known as hardening. Now you see stage 1 sterilization, stage 2 food production. So this is the steps, and then stage 3 pre-transplanting means rooting, means development of root, and then transfer to natural environment, that is known as hardening. I am repeating the word hardening. Hardening means shifting from lab to glass house, first greenhouse and then glass house. Gradually they get development of root and stems. Okay, so this is the steps for micro propagation. Now plant tissue culture of Nicotiana tabacum, that is the shoot generation, root generation. You can find the callus formation, then stem formation, then root formation, that is the steps. So advantage of tissue culture over the intact plant, this you can produce any chemical compound inside the bioreactor. Means if you through protoplast culture you can obtain the protoplast. आपको पता है प्रोटोप्लास्ट क्या है? प्रोटोप्लास्ट क्या है, कोई बता सकता है?
Out cell wall.
Without cell wall means you multiply that type of cell inside the plant tissue culture media. Okay, now the production of diploid plant from haploid plant shorten the time taken to achieve the uniform homozygous line. means आपको कम टाइम में ये homozygous line मिल जाएगा, आपको जिस वैरायटी का चाहिए वैसा वैरायटी आपको थ्रू प्लांट टिश्यू कल्चर मिल जाएगा. अगर आप चाहते हो टू डिफरेंट वैरायटी है जिसका नॉर्मल में जो सेक्सुअल मेथड में क्रॉसिंग नहीं हो रहा है, उसको आप प्रोटोप्लास्ट फ्यूजन से पॉलीइथिलीन ग्लाइकोल, इट इज अ केमिकल दैट यूज फॉर फ्यूजन ऑफ टू डिफरेंट प्रोटोप्लास्ट. मींस फ्यूजन ऑफ टू डिफरेंट प्रोटोप्लास्ट मींस फ्यूजन ऑफ न्यूक्लियस एज वेल एज फ्यूजन ऑफ साइटोप्लाज्म. साइटोप्लाज्म में भी जीन होता है. जीन कहाँ होता है पता है आपको? इनसाइड द क्लोरोप्लास्ट इनसाइड द माइटोकॉन्ड्रिया. इसके अलावा जीन मेन कहाँ होता है? इनसाइड द न्यूक्लियस. सो दिस प्रोसेस दैट इज known as hybridization, fusion of two different type of protoplast of two different type of variety in presence of polyethylene glycol and calcium ion is known as hybridization, and product is hybrid. हाइब्रिडाइजेशन क्या हुआ? एक का प्रोटोप्लास्ट लेंगे और एक का लेंगे प्रोटोप्लास्ट विदाउट न्यूक्लियस. प्रोटोप्लास्ट विदाउट न्यूक्लियस, तो उस कंडीशन में क्या होगा? न्यूक्लियस एक का होगा लेकिन साइटोप्लाज्म टू डिफरेंट वैरायटी का होगा. उस प्रोसेस को कहेंगे हाइब्रिडाइजेशन. सो वी कैन प्रोड्यूस द क्रॉस वैरायटी ऑफ द प्लांट थ्रू हाइब्रिडाइजेशन. हाइब्रिडाइजेशन in presence of polyethylene glycol, calcium ion of the two different protoplasts, that is for getting different variety. Cell selection increase the potential number of individual in the screening program. Means we can increase the large number of the plant from the each cell. Means after creating it a transgenic plant, that is the main advantage of that plant tissue culture. Micro propagation using meristem and shoot culture technique allow the production of large number of uniform individual of species from the limited starting materials. Genetic transformation of the cell enable very specific information to introduce the single cells which can be regenerated. Means if you want to do any change, you can do it. Okay. So now try to know. You can create a transgenic plant and then you can create a variety with the help of plant tissue culture. You can do the genetic manipulation inside the protoplast easily. Protoplast cell without cell wall, because there is no cell wall, and you can change the gene inside the chromosome, inside the DNA, inside the chromosome. Gene is present inside the DNA and DNA is present inside the chromosome, and there is no barrier in case of protoplast, because cell wall is the most important barrier for transferring the gene. So try to know what is the plant tissue culture, what is the micro propagation, what is the embryo rescue. Try to know the concept, and how can you produce the virus free plant, why we use the meristem for production of virus free plant, what is the secondary metabolite, and what is the advantage of that secondary metabolite for plant as well as human beings. So today you have to go through these slides and try to understand the concept. I have already discussed these things, application of plant tissue cultures. Okay. So this is the application. First production of secondary metabolite through creation of transgenic plants. Means you can get saponin or vincristine, vinblastine, and then large number of plant producing large number of that second. Then production